Hello, everyone. Welcome to this first lecture of LBT 280. Um, this is, we call this class generally urinalysis. However, we're going to do a few other things besides uh, urine testing. However, so we will start off. I'd like to do, first off, a brief lecture about the urinary system. I'd like to do that so that we can then move on Wednesday to one more lecture on urinary diseases and disorders. And from there, go on to do the lab. Um, the class will be composed of the usual structures, uh, discussions, um, lectures, an assignment each week, and an assessment. There will be quizzes, various quizzes that we'll have. There will be part of your hybrid material will be to watch other videos and take short quizzes as a result. There will also be a video on the renin, angiotensin aldosterone system, which is an important part of the hormonal system that is governed in the urinary system, specifically in the kidneys, involved in regulation of blood pressure. So you'll do that online, offline as well. So I'm going to just swing into this urinary system lecture, and I'm going to start by actually downloading this, which you can do as well, and take a look at it. And what we will do is go through this system briefly, and go on to talking about what else goes on in the class. So the urinary system is a place that where urine is extracted from our circulatory system, stored, and then has mechanisms for excretion. The urinary system helps in a number of different ways. Regulating electrolyte balance. Electrolytes are things like sodium and chloride and potassium and calcium and all sorts of chemical elements. Regulating pH. pH is a measure of acidity. The pH of our blood is in the range of about 7.35, which is slightly basic. pH of 7.0 is neutral, neither basic nor acidic. And anything under 7 is considered acidic. Anything over 7 is considered basic or alkaline. And this has to do with the ionization of water, mainly in the body. And the function of this is that this affects many physiological, biochemical, as well as chemical processes. Osmotic balance is the ratio of the various elements: sodium, potassium, chloride, carbonate, calcium, and a number of others. And of course, it aids in controlling blood pressure, as I mentioned before. There are a number of hormones that are related to urine excretion. They also help other bodily processes. The major hormones are the antidiuretic hormone. This is what keeps us from peeing too much. Aldosterone, something called natriuretic hormone, which natrium is sodium, so it's part of this the sodium balance, and renin. The antidiuretic hormone is also known as vasopressin, and vasopressin is released when the body becomes dehydrated and causes the kidneys to conserve water as opposed to continuing to dehydrate you. And the it's involved in blood pressure as well. Constricting vessels actually changes blood pressure. Aldosterone is a sodium-potassium pump. Conserving sodium, secreting potassium, helps in water retention, and by that increases blood pressure by increasing blood volume. ANH, or atrial natriuretic hormone, is released when blood pressure is too high. It inhibits water reabsorption. Kidneys will process water and reabsorb it, and by it's a natural diuretic in its activity, and it helps us get rid of excess sodium and thereby help control blood pressure. Renin is a major regulator. This one is actually made in the kidney in response to low blood flow, meaning low blood pressure. It leads to the formation of angiotensin. Angiotensin is a vasoconstrictor. In other words, it makes the arteries narrow. That raises blood pressure. And it increases aldosterone clearance and increases the retention of water and salt, again, part of controlling blood pressure. This graphic, which I urge you to go back through and look at later to understand how this works, the renin angiotensin aldosterone system. We get angiotensinogen is a precursor, which is processed by renin to become angiotensin 1, which is then enzymatically modified to angiotensin 2. And either this causes a constriction of vascular smooth muscle, and the stimulation of aldosterone, which increases water retention. And so we have changes in blood pressure that result from that. So this is a graphic of the system, various components, the kidneys, the ureters leading to the bladder. You can see where they're located. Kidneys are located on the back abdominal wall. We have obviously two of them. The structure of the kidneys are in broad terms can be listed here. So blood flows into the kidneys, and waste products are removed, made into urine, and secreted into the ureters to travel to the bladder. So we have kidneys, two ureters, one from each kidney to the bladder, and then one urethra from the bladder that allows the urine to be excreted from the body. Okay, so some of this information is A and P. It's a good idea for you to go back and review the A and P section. Go back and review your notes from A and P about the urinary system. The various terms here. Kidney is roughly the size of a fist-sized bean in shape. The hilum, an indent that faces the spine. This is the entry and exit point for major blood vessels, as well as the ureter. The outer portion is the cortex, which surrounds the inner portion, which is the renal medulla. So here's another example, a little more structural here. And we have papillae are like fingers. The renal capsule is incorporating it, the ureter, artery coming in, vein going out as usual. And we have the medulla and the cortex as well. You should remember that from your A&P. Renal pyramids are within the renal medulla. They're just because of their shape. Cortex extends between the pyramids to form columns, and the cortex and medulla, they contain the nephron, the functional unit of the kidney. Ureters carry the urine to the bladder. They are in the range of a foot long in an adult. Gravity and peristalsis, meaning sequential squeezing of the muscles, the smooth muscles that surround the ureter, move the urine into the end of the bladder. And there are valves that keep it from backflowing as well. If we look at the ureters, there's connective tissue, muscles. One of them wraps around the ureter, the other runs the length. The inner layer secretes a mucus to protect that lining. And the lining, it's important to protect the lining from a variety of things. So the bladder is a sac, holds between 1/2 and 3/4 of a liter. There are 3 layers in the walls. The inner layer, the mucous membrane, is continuous with the ureter lining, meaning there are no seams. So basically, it's a development in terms of developmental biology. It comes out of the development of the system as an extension of the ureters. The rugae are in the bladder just the same as they are in the stomach. They're basically folds. The bladder is not is not full of air. Shouldn't be ever. Therefore, it's expanded to its capacity when urine flows into it. So if we look at this, the rugae are here. We talked about here comes the ureter. The lining of that is contiguous. We have the trigone, which is basically a triangular shaped space, connective tissue, mucosal layer, a submucosal layer, and then what we call the detrusor muscle, which allows the thing to be contracted and push urine out. You've got the ureter comes in, there's an opening, and then there is a urethral valve as well down here that goes to the extern. And in men, there's a prostate gland, obviously in women, not. The bladder is covered with a membrane, the parietal peritoneum. The brain signal, when we are urinating, the brain signals that detrusor muscle to contract. In other words, it contracts on the bladder and pushes, puts pressure on the urethral sphincter. So the brain signals the external sphincter to relax, and then the urine flows through. Let's do this. The trigone or trigone is a triangular area made of the urethral and ureter openings. Tissue flaps cover these things to prevent backflows, much like our heart valves. So the urethra carry urine from the bladder to the outside. In men, this is 6 to 8 inches long. In women, it's shorter for obvious reasons. Sphincters control that. There are the internal sphincter is not under voluntary control in general, so it opens when the bladder is stretched. We can keep the external sphincter closed. So what happens is we trigger the urinary system works by filtering waste out of the plasma. So we have a couple of we have a couple of terms that we want to throw out. As an adult, depending on the amount of fluid intake, you push out 1 to 2 liters of urine a day. In general, for good kidney health, you want to be more toward the 2-liter end because it helps flush out all sorts of waste materials. Polyuria is more than 2.5 liters a day, poly meaning many. Oliguria, less than half a liter, less than 400 milliliters a day. And anuria means basically no urine, less than 100 milliliters a day. So to make urine, we go through several different processes: filtration, reabsorption, and secretion. So waste is left in the blood and bowels, and our cells contribute other waste products of metabolic activity. All of these various parts of the body secrete waste. And in the urinary system, in the lungs, the skin, through sweat, and the intestines all do this. A major component of urine, and why we call it that, is urea. And this is done when we eat a lot of protein. The nephron is the basic functional unit. Kidneys have about 1 million of these things. They are a glomerulus and a set of tubes. The glomerulus is in the Bowman's capsule structure, which is within the cortex. And the renal tubules, the tubes that go out of that flow back and forth, flow into and out of the glomerulus, are in the medulla. The structure of the nephron is fairly complex. And again, this is something you should go back and spend some time reviewing. We have in the nephron, we have the glomerulus, which is this structure where the blood flows into and then out of by a couple of arterioles. And then this is where the blood is filtered to produce the beginning part of the urine process. It moves into the first loop here, the proximal convoluted tubule, where reabsorption of water, ions, and other necessary nutrients begins to occur, moving in then to the next part of the renal tubule, the descending limb, into what is known as the loop of Henle, where further reabsorption of water and occurs. First, the water is absorbed, excuse me, in the proximal loop, the descending loop, and then the ascending limb of this loop. Other sodium and chloride and other components are absorbed, and this continues through this several portions here to get to the distal convoluted tubule. Distal convoluted tubule is called because of this convoluted shape, where we can get secretion of various things: acids, drugs, toxins, variable absorption, reabsorption of water and sodium, controlled by various hormones that we've discussed earlier. And then the final part of the nephron is the collecting duct, where things can come in and go out. Water can go out, solutes can come in, go out, and then then the papillary duct delivers urine to the minor calyx, and that structure at the in the little curve in the kidney to go to the ureter. So this is a complex biological process that is well regulated by the renin-angiotensin-aldosterone system as well. The renal corpuscle is located right when we go to the nephron. So we talked about the structures here. Bowman's capsule is a sac that surrounds the glomerulus. So we're looking here again. This is a reverse image of what we looked at before. So the arterioles come in and go out here, and then we have the processing going on and heading into the Henle loop, and the proximal convoluted tubule. This capsule here is a very thin layer, but it does surround the entire glomerulus. So we talked about that. Blood pressure forces the small molecules to pass out through those capillary walls in the glomerulus. The arterioles come in, and then they become shrink down to capillaries, and they then can pull out glucose, ions, urea, and blood pressure is the force that carries them through. Podocytes are cells that have the ability to direct filtrate into space, into the glomerulus and capsular space. We talked about proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Proximal means closer to the glomerulus. Distal is on the other side of the loop of Henle. And then there are all sorts of capillaries that surround the tubules. So again, if we look again, this is a different view. We have the glomerulus in the capsule here, and we have the proximal tubule here, where material goes in, circulates around, drops into the loop of Henle, and it's surrounded by capillaries. We have arterial capillaries. Okay, so blood comes in from the renal artery, filtered. The process starts here with the in the glomerulus. The blood then comes out and surrounds all of these tissues. It's still supplying oxygen to these tissues, and now that's venous blood heading back to the renal veins. So it goes into the proximal tubule and into the loop of Henle, and then into the distal tubule, and finally into a collecting duct. So the collecting duct has all sorts of little entrances from a variety of nearby nephrons, and then so there are a number of these as well that then come along and get to and go finally heading toward the ureter. Just like the digestive tract, the proximal convoluted tubule has villi. In other words, little fingers that extend out into this to increase the surface area. This is basically the longest part of the tubule and does a lot of the reabsorption work, various things. There are some issues here, something we call the renal threshold. Um, the if the um, if materials that are supposed to be being reabsorbed are too high, like sodium and potassium and chloride and a few other things, then it doesn't get reabsorbed and ends up in the urine. The glucose threshold is 160 to 180 milligrams per deciliter. Remember, a deciliter is 100 milliliters. And diabetics, the reason that we see glucose in the urine of diabetics is that they have levels above um, the 160 to 180 milligrams per deciliter. When we see this increase in sugar that we can measure in the urine, it's called glycosuria, and it's one way of actually looking at and diagnosing diabetes in patients. From the proximal tubule, convoluted tubules that goes into the loop of Henle, we have descending and ascending loops, and then and actually they're just corridors basically, and then the actual loop is where it changes direction. So the descending loop is quite permeable to water, so it will not take electrolytes. The ascending loop is somewhat permeable to water, but very much to potassium um, and chloride ions. And then reabsorption of ions occurs further up the ascending loop, and it's reabsorbed by what we call active transport, meaning that there are molecules in the system that act as transport molecules to facilitate that reabsorption. This is as opposed to simple osmotic pressure, meaning too high a concentration and it will come out. So active transport means basically it goes against the direction that they would go and what we call the concentration gradient. Biological systems and chemical systems have a tendency, we call it osmotic pressure. If you have two regions of which contain a concentration of, say, sodium and a permeable membrane between them, then they will tend to equilibrate. In other words, sodium will migrate from the higher concentration area into the lower concentration, or water will migrate from the lower concentration area into the higher to dilute it. Anyway, there's two ways that can happen. Active transport actually makes things go. This water moving from dilute to more concentrated areas is the normal method of osmotic control. Active transport actually moves things in the other direction, against that gradient. Okay. In the renal tubules, we have the antidiuretic hormone, which controls the amount of water reabsorbed. If we have more salt in the blood, more ADH is made. If we have less salt, we have less ADH. And the kidneys will adjust to dietary insult, or dietary intake, rather. And then you'll see patients on kidney dialysis. This is what happens when the kidneys are failed. In other words, there is sufficient damage, chemical or environmental damage to the kidneys, or maybe they're just gone, that we need to have a machine that will actually remove these waste products. The distal convoluted tubule is on the far side of the ascending part of the loop of Henle, and these cells work in terms of active transport to pump harmful substances into the urine. This is where drugs get pumped into the urine. This is not just illicit drugs, but the kidneys are the main excretion method for drug metabolites and things that don't belong in the body. Ammonia is a basic substance. It tends to raise pH. The body tends to buffer that as much as possible, and one means is pushing ammonia out. Urea is a nitrogenous compound that comes from the metabolism of proteins. And then we get a lot of reabsorption of water and secretion in this case of potassium. Potassium sodium balance is critical to proper health, so the kidneys are involved in that balancing act. Collecting ducts collect the filtrate from a lot of nephrons, and the cells in the collecting ducts adjust their permeability to water controlled by ADH. The tips of the renal pyramids are called papillae. These papillae are like fingers, and some drugs that have an effect on kidneys will actually affect this area. This is misspelled. It should be. There we go. NSAIDs, like non-steroidal anti-inflammatory drugs like aspirin and ibuprofen cause problems here. Ibuprofen is much harder on kidneys than aspirin. ACE inhibitors, angiotensin converting enzyme inhibitors, work here as well. They are an important part of the blood pressure control armamentarium, but they also have this. Amphotericin B is an antifungal drug, a particular nasty drug. The collection ducts, so we go from the tubules to the minor calyces, to the major calyces, and then to the renal pelvis. And then the renal pelvis is this area that directs urine into the ureters and then on to the bladder. Flow rate is important. The filtration rate, glomerular filtration rate, is a measure of kidney activity. It's basically the amount of filtrate that is generated through the kidneys in a minute. So the kidneys can process up to 180 liters a day. Now remember, a lot of this gets reabsorbed. In fact, 99% of it is reabsorbed. But urine consists mainly of water, like almost everything else, but in this case, much higher percentage. So about 96% is water, and the rest is divided between urea and the metabolic products that are taken care of. So in urinalysis, we look at a number of different things. Concentration of various components of urine, and metabolic products, and the presence of cells, crystals, and other abnormalities. And we'll be going into that later. Okay, so let's do this. I would bet that that the PowerPoint didn't come through. Apologize for that. I'm going to suggest that you pull up that PowerPoint and follow with me during the lecture. Okay. Let's go back now to week 1. So again, please look through the PowerPoint and follow through. I've gone through it fairly quickly. On Wednesday, we'll look at urinary diseases and disorders, and we'll be doing some work in the lab as well. Get started on a couple of things. We've got we have a drug screen quiz. So I want you to do this before Wednesday. So you're going to click here to read instructions for the test. This will take you to accutest.net and then read through and then take the test at the end. Take a screenshot of that and submit it for credit on this particular quiz. We'll be doing the dipstick urinalysis and drug screen and the HCG. HCG is human chorionic gonadotropin, which is a pregnancy test. It's what the doctors will use when they do that pregnancy test in the office. Hopefully, none of you will come up with a pregnancy in this class, although we've had some people get that happy news, surprisingly. But anyway, that's what we'll be doing this week. Your other parts, you can get started reviewing the videos. If you look here at RAAS, the renin-angiotensin-aldosterone system, you'll watch this video, and then you will, we go back now, you'll take a quiz, and then we have 2 other videos that you can watch as well. This will help you get prepared for your assessment for the week. Let's look at the discussion here. You can get thinking about this. We're looking at various diseases and disorders. What I'm going to modify this to a slightly different list and let you and actually change a couple of these things, we'll go from there. So this list will be a little different when you see it in the shell. The assignment for this week really is to write a paper of at least 300 words on the structure of the urinary system, the A and P of the urinary system. So that's another good reason to go back and take a close look at the PowerPoint that it looks to me like I may not have gotten that done for you, and I apologize for that. So go ahead and get started on that as well. I'm urging you to do that because this is going to be a busy class. You can email me. My email address can be found in the instructor contact information tab. I will be on campus all day Wednesday. If you want to come in early to talk to me, we will meet in LC1 and move to the lab as appropriate for each of our classes. As I said before, this is a busy, busy class. There's a lot of activity in the lab, so please don't miss class. And please stay up. We find a very direct correlation between attendance in class and keeping up with the work and succeeding in the class, getting a decent grade in the class. The fastest way not to pass a class, especially a class that involves a lot of lab activities, is to miss class. So please do whatever you need to get in to class. And I will see you all on Wednesday, and I hope you enjoy your Labor Day holiday.